Well, let us talk about we have the DNA sequence which is coming to us from different resources or from different portals, and the important thing is how we are going to use this DNA sequence, which could be only few hundred base pairs, or which could be million and billions of the base pairs. What are the important issues which we need to discuss before even thinking to study this DNA sequence? There are two very valid and important point. Number one, this DNA sequence is so extensive, it's so high throughput, it's so high density, it's so high regulatory that in order to localize it to be generalized, we have to consider for sure that over exempted data of this DNA sequencing could be much and much better than the underrepresentative data. Now, something more important to consider, first of all, 2 points to be considered and put in the mind of each and every one of you. Number one, we need a system, we need a way, a pattern, a method which is required to represent this DNA sequence in a way which could be acceptable, which could be appealing, which could be beautified for the person to look into. The person says, wow, now this is the sequence I want to read, that looks very nice. This ATGC, this GTAC looks very nice now. Either DNA or either the messenger RNA or any other source, or from any other source. Number one. Number two, well, the representation is already there, but the issue is this is a representation of the DNA sequence, or in a simplified way, this is only a DNA sequence, a generated or derived or annotated DNA sequence. Now, what else we need? We need to specify, we need to identify, we need to link it, we need to group specific areas within this DNA sequence to be considered as the functional one. Like for example, base pair number is, if it's for example 400 base pair sequence which is given to any of you, and we ask you, like, out of this 400 base pair sequence, what is the importance of this 400 base pair sequence? You could find what are the STS in the sequence, you could find what are the ESTS in the sequence, or you can come to me and say these are the transcriptional factor binding sites, there are different elements found in this system. So all these specific elements, what are the elements? Elements are the specific regions in the DNA sequence, or different regions of interest, or different functional regions of interest within the DNA sequence. So to map or to locate or to identify these specific elements on the DNA sequence which has been given to you is called DNA mapping. So please don't confuse DNA mapping with DNA sequencing. Both the terms are a bit different. In fact, based on these terms, if you are not sure, then you get more bigger issue when you're going for the DNA counting or DNA assembly. That is something different. But yes, DNA mapping is entirely different things. You got three basic types of DNA mapping. You got the cytogenetic mapping, you got the genetic linkage or genetic mapping, and then yes, genetic mapping is also called the meiotic mapping, and then the third one, which is basically we call the physical mapping. But yes, we are not going to talk about the subtypes, but the most important thing which you need to keep in your mind, there is a difference between the DNA sequences and the DNA mapping. Okay, well, now